Hi, my dear students. Myself, Anu Shashidharan, Academic Director and Biology Faculty of Rankings Entrance Training Academy. I welcome all of you in this video lesson. Today, I am going to talk about Biotechnology Principles and Processes, Part 2. Do you remember the last lesson we have discussed the topic recombinant DNA technology? And we have discussed the basic steps involved in this process. Okay, I repeat once again here. The first step, that is the isolation of DNA. Okay, you remember we have discussed the topic based on an example, that is insulin production. So the first step is the isolation of genomic DNA. Second, the fragmentation of DNA by using restriction enzymes. Third one, the screening the DNA fragments and identifying the desired DNA by using a technique named gel electrophoresis. Yes. Then insertion of foreign DNA into a vector DNA that leads the formation of a recombinant DNA. Fifth one, introducing that recombinant DNA into a host cell. We remember, as per our example, E. coli cell as the host one. Yes, and again, culturing the host cell in a medium at a large scale. And the last step, you are very familiar, obtaining the foreign gene product. As per the example, you remember what is the foreign gene product? Human insulin. Today, in this video lesson, I will discuss the first two steps, that is the isolation of DNA and the fragmentation of DNA by using restriction endonuclease enzyme. Okay, first of all, I'm going to talk about isolation of genetic material. As you know, DNA as the genetic material in most of the organisms. Yes, you are very familiar. DNA actually situated inside the nucleus. Suppose you think about this is actually a cell. This is the nucleus. The DNA actually present inside the nucleus. So for achieving the isolation of DNA, we have to treat the cells with the various types of enzymes. Okay, the isolation of genetic material can be achieved with the help of certain type of enzymes. Suppose if you want to isolate the DNA of a bacterial cell, you have to treat the cell with the lysozyme enzyme. Is it clear? Okay, if it is a plant cell, you have to treat the cells with the enzyme called cellulase. If it is a fungus cell, you have to treat it with a chitidase enzyme. Okay, then another important thing you are very familiar, inside the nucleus, the DNA actually intertwined with a certain type of proteins. I hope you have learned in class +1. The proteins are called histones. If you need the pure form of DNA, we have to separate all other macromolecules or all other biomolecules present inside the nucleus, especially the RNA and the proteins. Okay, finally, what we have to do, we have to treat it with the ribonuclease enzyme. As a result, the RNA can be removed. And again, you can treat with the enzyme called protease for the removal of protein. Is it clear? At the end of the process, we have to add chilled ethanol. As a result of the entry of chilled ethanol, what happened? The DNA molecule should be precipitated in the form of thread-like structures. Is it clear? This is also the diagrammatic representation of a precipitated DNA after addition of chilled ethanol. Okay, this is actually the precipitated DNA in the form of thread-like structure. And finally, we can remove the DNA. Okay, the process is called spooling. As a result of spooling, what we will get? We will get the entire DNA of the cell. Okay, as per the study, you remember, we have to produce human insulin by using rDNA technology. If it's so, we have to isolate the DNA from a human cell. But at this time, we are not able to identify which one is the desired DNA or which one is the DNA responsible for the production of insulin. We got the DNA as a whole. So clear, we got the DNA, I mean the entire DNA from the cell. Okay, at this time, we are not able to identify which one is the desired DNA. Okay, this is the different steps in which the isolation of genetic material. So at the end of this step, what we get? We will get the entire DNA of the cell. This is the first step of recombinant DNA technology. Is it clear? Second one, you are very familiar. We have to cut the DNA molecules into fragments. Okay, for cutting the DNA molecules into various fragments, we are using restriction enzymes. Restriction enzymes are commonly called molecular scissors. Okay, now we are going to discuss the detailed aspect of restriction enzymes. Is it clear? Restriction enzymes, it's a group of enzymes which are basically perform the cutting of DNA. Okay, the fragmentation of DNA at a specific site, not for all sites. The restriction enzymes can cut the DNA at a specific site. Okay, the restriction enzymes actually belongs to a large group of enzymes called nucleases. It's a large group of enzymes. Nucleases are two types. See, nucleases are two types. First one is called endonuclease enzyme and second one is called exonucleases. What is the meaning of exonucleases? Exonucleases can commonly using for removal of nucleotide from the end of the DNA. That means the exonucleotide should not cut the DNA both of the strands. It should only remove the nucleotides from the end of the DNA. It's clear? But endonucleases, it makes cuts at specific positions within the DNA. That you will be clear in the next session. Okay, endonucleases, it can make cuts at specific positions within the DNA. It means endonucleases can be able to cut both the strands of DNA. You are very familiar. DNA is actually having double helical structure, isn't it? The detailed aspects we are going to learn now. First of all, we have to learn the nomenclature of restriction enzymes. Dear students, there are so many restriction enzymes we have already been identified. Around 900 restriction enzymes had been obtained from different types of prokaryotic cells. Did you clear? That means the restriction enzymes can be obtained from certain prokaryotic cells, especially bacteria. One of the most important restriction enzyme is called EcoR1. Okay, this is actually the most important restriction enzyme. Second one we have to learn, it is called Hind second. Hind second is the first isolated or first identified restriction enzymes. Okay, now we have to learn the nomenclature of this type of restriction enzymes. First of all, I'm going to discuss EcoR1. I said the naming of the restriction enzymes based on the name of the prokaryotic cells from which it has isolated. For example, EcoR1, the restriction enzymes obtained from a bacterium called Escherichia coli. I hope you might hear about it. It's commonly called E. coli bacterium. Its full name is Escherichia coli, isn't it? Now I am going to discuss the nomenclature of restriction enzymes. Look, the first letter of the restriction enzymes, that is, came from the first letter of the genus name of the prokaryotic cell. Okay, the prokaryotic cell is Escherichia coli. Escherichia as the genus name and coli as the species name. So here you can see the first letter, that is the first letter of the genus name of the prokaryotic cell. The second and third letters of the restriction enzymes came from the first and the second letter of the species name, CO. Okay, here the R means, the third letter R means it's from the name of the strain. You know, there are different types of Escherichia coli bacterium. Out of so many types, now the most important strain called RY13. Okay, so this R came from this RY13. And here the 1, 2, 3 like that. That is the order of discovery, means the order of isolation. Is it clear? This is called a Hind second. You know, the Hind second enzyme obtained from a bacterium called Haemophilus influenzae. So you can identify how the nomenclature occurs. The first letter came from the genus name of the bacterium. The second and third letter from the species name, the first and the second letter of the species name. And the fourth letter came from the name of the strain. The strain is called the RD. And here the second means, or two means, order of discovery. I hope you have understood the nomenclature or naming of restriction enzymes. Okay, now we are going to learn in detail the action of restriction enzymes. Dear children, the restriction enzyme should not cut at all the regions of DNA. The restriction enzymes can make cut at the specific regions of DNA. Such specific sites are collectively called a recognition sequence or restriction sites. Look, this is actually the restriction site of a particular type of restriction enzyme called a EcoR1. This is the restriction site of EcoR1 enzyme. Such type of recognition sequence or restriction sequence always having 4 to 8 nucleotides. Okay, your NCERT book saying that 6 to 8 nucleotides. Here you can see 6 nucleotides are there. G, A, T, T, C. Is it clear? And another important characteristic feature, all type of restriction sites are palindromes. Such type of sequence are called a palindromic sequence. Do you know what is the meaning of palindrome? You are very familiar. A palindrome is a word, phrase, verse or sentence that read the same forward or backwards. Look, the most important word, Malayalam. It's actually a palindromic word. If we are actually reading from left to right or from right to left, we can read the same way. Such type of words are collectively called the palindromic words. Likewise, all type of recognition sequences are palindromes. Look, suppose you think about this is actually a DNA fragment. If the DNA actually cut like this, which strands are moving right side and left side? The strands move left side, that having the base pairs A, T, T, C. Look, here also you can see A, T, T, C. Such type of sequences are collectively called palindromes or palindromic sequences. Listen, dear children, this is actually the diagrammatic representation of fragmentation of DNA by restriction enzymes. Suppose you think about it, this is actually a double-stranded DNA. I said the specific sequence in which the restriction enzymes can make cuts, that particular sequence we can call it as recognition site. Okay, this is actually the recognition site of EcoR1 enzyme. Look, the way in which they are cutting the double-stranded DNA. Look, this is the way in which the cutting of DNA. As a result, what happened? Two strands will be there. Two complementary strands are there. Look, in this type of cutting, you can see both the strands are actually having some overhangs. Okay, the overhangs are there. Such type of ends we can collectively call the sticky ends. Okay, as a result of cutting of certain restriction enzymes, they can produce sticky ends. Sticky ends means the end of the DNA actually having complementary overhangs. Such type of ends we can call it as sticky ends. The sticky ends are suitable for recombinant DNA technology. In some other type of restriction enzymes, they can make cut like this. Okay, as a result, what happened? The two ends, I mean the two fragments, they are having uniform edges. They are not having overhangs. Okay, such type of ends we can collectively call the blunt ends. So, as a result of cutting, what happened? The DNA molecules, I mean the fragments may be produced either blunt ends or sticky ends. Okay, the sticky ends are suitable for ligation. You remember ligation that we have discussed last day. Okay, the joining of vector DNA and foreign DNA. Okay, that process is called ligation. The ligation, for the process of ligation, this type of sticky ends are suitable. Okay, this is actually a table. It's showing so many enzymes. Okay, it's source organisms, its restriction site or recognition sequence, and structure of cleaved strands. That means the result of cleavage. Cleavation means the cutting of DNA. The first one we have already been discussed, EcoR1. It's actually obtained from a bacterium. You remember, Escherichia coli. What is the recognition site of Escherichia coli? GAATTC. That means, dear children, if the DNA molecules wherever having that particular sequence, what happened? The EcoR1, such type of restriction enzymes can make cut at this particular site. As a result, what happened? Two sticky ends are formed. Okay, for plus 2 students, you have to learn only one enzyme, that is EcoR1, its source organism and its restriction sites. Those who are preparing for NEET or any other national level entrance examination, you have to read, you have to learn so many other types of enzymes. Okay, for them, I'm actually going to discuss few more restriction enzymes and the source organisms and restriction sites. Second one, PST1, is actually obtained from a bacterium called Providentia stewarti. Okay, its restriction site CTGCAG. As a result, what happened? It also produced sticky ends. Third one is called SMA1, SMA1. Okay, its source organism is a bacterium, Serratia marcescens. Its restriction site CCCGGG. But here you can see it's actually may cut like this. As a result, that creates two blunt ends, not sticky ends. Fourth one is the He third, HAE third. It's obtained from a bacterium, Haemophilus aegyptius. Okay, its recognition sequence, you know, GGCC. Okay, it also creates blendent. The last one, HPA second. It's obtained from a bacterium named Haemophilus parainfluenzae. Okay, its recognition sequence CCGG. So I say all type of recognition sequence or restriction in restriction site having 4 to 8 or 4 to 6 or 4 to 8 nucleotides are there. Okay. You have to learn it. And the last step I'm going to discuss the cutting and the formation of recombinant DNA. Suppose you think about it, this is actually a vector DNA. You remember vectors? As per the study, we are using plasmid as the vector. This is a plasmid DNA. Do you remember what is the meaning of plasmid? Plasmids are extra chromosomal, circular, self-replicating autonomous DNA molecules present in bacteria. Suppose you think about this is a vector DNA. This is a foreign DNA. As per the example you remember, okay, this is the foreign DNA, means the DNA obtained from human cell, okay, or the DNA responsible for the production of insulin. This is a foreign DNA. So we have to ligate these two DNA to form a recombinant DNA. So first of all, what we have to do, we have to cut both the strand of DNA by using the same enzyme. Okay, the enzyme called EcoR1 over here. As a result of cutting of vector DNA and foreign DNA, what we will get? We will get the sticky end. Okay, this is the one fragment of vector DNA. This is another fragment of foreign DNA formed as a result of fragmentation by EcoR1. You just imagine these two sticky ends are complementary. These two sticky ends are complementary, which are suitable for ligation. At the end of the process, what we have to do, we have to ligate these two DNA molecules. By using an enzyme, it is called a DNA ligase. Is it clear? So this is actually a recombinant DNA. That means the combination of two DNA. This is the vector DNA and this is the foreign DNA. Is it clear? So in this video lesson, I have discussed two important steps of recombinant DNA technology. That is the isolation of DNA and the cutting of DNA at a specific site by using restriction enzymes. I hope you have understood all the concepts. Thank you.